In this video, I will demonstrate how to run a workflow for bioinformatics analysis of SARS-CoV-2 Oxford Nanopore technology sequencing data. The workflow runs in the software Geneious Prime in its graphical interface. Therefore, it's a workflow that can be set up rapidly and no programming knowledge is required. The structure of the workflow is as following. The input data are FASTQ files generated from the Nanopore sequencing of your SARS-CoV-2 samples. The FASTQ files are trimmed. The trimmed reads are then mapped against a reference genome, either the one that we provide or a reference genome of your choice. From the alignment, a consensus sequence is generated. The consensus sequence is then annotated. We provide two databases with common mutations and other functional mutations, although the user is free to adjust or change the database, depending on what mutations to search for in their analysis. The workflow can easily be adjusted for genome analysis of other pathogens. This is the basic layout of our analysis workflow for SARS-CoV-2 sequencing data, and the workflow is called SARS-CoV-2 Assembly Basic. We have also created a workflow called SARS-CoV-2 Assembly Wrapper Plugins. It contains the same functions as the other workflow, but includes two additional steps. The software Pangolin and Nextclade are included as wrapper plugins, whereas Pangolin gives Pango lineage assignments. Nextclade gives clade assignments, mutation calling, and quality checks. Both workflows can be used with the Arctic primers or Midnight primers. The workflows are designed to work on Windows, Linux, and Mac. This video is a step-by-step guide on how to set up the workflows in Geneious Prime. I am using Geneious Prime 2020-321 on a Windows computer, but this tutorial is also aimed at those using Linux or Mac. To run the workflows, you'll need the software, Geneious Prime, which should also have the plugin Minimap 2 installed. I will show you how to check if the plugin is installed and how to obtain it if not. If you are planning to install the workflow SARS-CoV-2 Assembly Wrapper plugins, you will also need Python, Docker Desktop, used to obtain and run Pangolin, Nextclade CLI. Links for these software are in the description. The files required for the workflows are available at the Geneious SARS-CoV-2 repository at GitHub. Link is in the description. You can choose how you want to download the data, but I will copy the URL address and use git clone. The data is now downloaded to my computer. In Geneious Prime, we will now import the downloaded data files. Select in which folder you want to import the data into. I am using local. Then we go to add and import folder. Now you need to locate the Geneious SARS-CoV-2 folder you got from GitHub, and in that folder there is another one called SARS-CoV-2 Public Workflows. Choose that folder and click Select. You should now have the files in a folder structure looking like this. What we have imported are the sets of primers used for the trimming, the reference sequence used for the alignment, and the annotation features. Before we set up the workflow, we'll have a look to see that Minimap 2 is available in Geneious. You can see this if you go to Tools, Plugins. Under Installed Plugins, we currently can't see Minimap 2 there, so we go to Available Plugins, find Minimap 2, and click Install. We will now import the workflow. Select Workflows, then Manage Workflows. Click on Import, and then in the Geneious SARS-CoV-2 folder, we can see the two workflow files. Choose the workflow you want to import, and it should be visible in the Workflows list. I will import both workflows like this. Before we can run the workflow, we need to do some configurations. Select the workflow you are planning to use. I will open the SARS-CoV-2 Assembly Basic, but if you are using the SARS-CoV-2 Assembly Wrapper plugins, you can open that instead, as these instructions apply to both workflows. The steps that may need manual editing are trim using bbduke, align, assemble, map to reference, annotate from database, both of them. Export. We'll start with trim using bbduke. Double-click on it, and this window will appear. Select Choose. Now we need to find the primers we're planning to use. Here in the folder we imported, we got the Midnight primers and the Arctic primers. I will choose the Midnight primers. Click Use Folder. Once that is done, click on Apply and Close. Now we will edit the Align Assemble Map to Reference. Double-click. Next to Reference Sequence, we want to specify which reference we'll use, so click on Choose. Find the reference sequence. I will choose the one we downloaded from GitHub. Press Select. Then we want to change the mapper. Hopefully you will see Minimap 2 available. If not, you need to install the Minimap 2 plugin. Once this is done, we click Apply and Close. Next, we will specify the annotation features. You can see there are two steps here, both called Annotate from Database. We will edit both of them. We will start with the first out of these two. Next to Source, you can click and select the folder with the annotation features. In this case, the folder Common Mutations has already been selected, so there is no need for me to change anything. Then we repeat this in the second Annotate from Database step. Again, the folder with annotation features I want to use has already been selected, so I will leave it like that. Then we will edit the step called Export. In this workflow, it's the last step, but if you're editing the workflow with the wrapper plugins, there are 3 export steps that need to be edited. Next to Export to Folder, it's possible to specify where on your computer you want to save your analysis results. Geneious has already picked a default location, but I want my results to be located in another folder on my computer. So I click Browse, and here is a folder I named Results. I will select that one. Apply and Close. If you're using the basic workflow I've been editing, you can click on Apply and Close to save your changes and start using your workflow. You don't need to finish this video. However, if you are using the other workflow with the wrapper plugins, we need to add those plugins into the workflow. So we will download the plugins and import them into Geneious before doing so. Plugins for Pangolin and Nextclade are available in separate repositories on GitHub. You can find the links in the description. We'll start by going to the Pangolin repository. There are more instructions on this page on how to obtain and install the plugin, or if you want to create the wrapper plugin from scratch in Geneious. But in this case, I will show you the easy way to just download the G-plugin file and import it. As you can see, there are 2 of them, one for Windows and one for Linux and Mac. Since I'm using Windows, I will select the one called Pangolin Windows 1.0.g plugin, and I'll download it like this. Then I'll do the same for Nextclade. I will choose the G-plugin file for Windows and download it. Now the plugins are ready for installation. In Geneious, click on Tools and Plugins. Click on Install Plugin from a G-plugin file. Here are the G-plugin files I just downloaded. I'll start with Pangolin. Select. Install Plugin. OK. Again, the plugin should be visible under Installed Plugins. Now we'll do the same for the Nextclade plugin. Next, we will add the plugins to the workflow. Open the workflow in editing mode. The Pangolin plugin should be added here. The Nextclade plugin should be added here. We'll select this step, so the plugin will be placed under it. Go to Add step. Then we can search for the Pangolin plugin in Add Operations. I will search for Pangolin, and there it is. Select it, and now the plugin is in the workflow. We will do the same for the Nextclade plugin. Select this step, Add Operation. And there we have the Nextclade plugin. Finally, we need to specify some paths in the added plugins. We'll start with the Pangolin plugin. Double-click on it. We need to fill out the path to your Docker desktop and the path to your Geneious data. I will start by adding the full path to where my docker.x is located on my computer. The Geneious data path is a little trickier. On your computer, there is a folder for all the Geneious Prime data. On my computer, it's in the home directory, And the folder is called Geneious 2023.2 Data. In this folder, there is another folder called Transient. This is the Geneious data path, so I will copy the path and add it. Apply and close. Next up is Nextclade. Double-click on this step. We need to add the path to where your Nextclade is located and also the destination where you want to export all your Nextclade output files. I'll start by adding the path to my nextclade.exe file. Then I'll add the location for my Nextclade results. I'm using the same folder as where the other results files are also exported to, the one named Results. Apply and close. I will also make sure that all export steps are linked to this results folder. Click on Apply and close. Now your Geneious workflow is ready to run.